discuss my work. I will try to, the talk I prepared is modular, so there are modules. At the end of each module, there is a little summary. So I hope we can go through a few of them, and please feel free to interrupt at any time and ask questions. I will be happy to discuss. So the work I will present is, I mean, you can find it in these 3 papers, and it has been done in collaboration with these people here. We have a joint group running between Rome and Paris. So Martin White is my colleague in Paris who is running this group with me. And we have a GitHub where you can find all our codes. So I mean, if you need any of these codes, please check the GitHub, and we are happy to talk about the codes and how to use them. Now, what is the goal of our work? So we work on proteins, and as you, I guess, all know, in protein, I mean, in biology, I mean, the main paradigm about how protein work is that you have proteins are machines that have to perform some function, and proteins are also, physically speaking, they are chains of they are polymers, so they are chains of amino acids. So you can represent them as a sequence. So you have a sequence of amino acids. Each amino acid is represented by a letter. And then the idea is that this sequence, that is a chain of amino acids, is going to fold into a complicated 3-dimensional structure. So you have a structure. And then this structure will allow the protein to perform some function. So for example, there will be some binding site, there will be some part of the structure that can move, and so on and so forth, to perform the function that you want to perform. So you can move, in principle, from sequence to structure to function. Now, the problem of predicting the structure starting from the sequence, as you all know, I guess, has been solved essentially by AlphaFold and similar models. The reason why it was possible to solve this problem is that we have a lot of data. So we have about 10 to the 8 sequences of proteins that you can get from public databases, and we have about 10 to the 5 structures that are also available thanks to experiments that have been done. So you have a lot of data to train machines, and so people were able, thanks to this, to develop, I mean, these modern deep networks to train the machine to predict the structure given the sequence, and then use it to predict a new structure. So this was very successful. There was a Nobel Prize for it. And the other thing about structure is that, mathematically speaking, structure is a well-defined quantity, because you can, it's a physical property of the protein. You can give the coordinates of the atoms in the 3-dimensional space, and this is very well defined mathematically. So it's easy. I mean, it's relatively easy to, I mean, it's easy to formalize the problem, and then you can proceed to attack it with these modern techniques. The problem when you want to predict a function is much more complicated for 2 reasons. One is that we have less data, so there are not so many experiments that are able to measure the function of a protein. For example, a typical protein I will be looking at is what is called beta-lactamase proteins. These are proteins that are used by bacteria to protect themselves from antibiotics. So when an antibiotic arrives, it will attack the bacteria, but the bacteria can produce this protein, and the protein will bind to the antibiotics and cut it, so to neutralize it. Now, so this is the function of the protein, but the question is, how do you measure the function? So typically what people will do in an experiment is to grow the bacteria with a certain amount of antibiotics, and they will measure the growth rate or something like this. But of course, this depends a lot on the experimental conditions, so the environment, the amount of antibiotics, and so on and so forth. So measuring functions, I mean, expressing functions in terms of a few numbers is complicated. I mean, function can be a bit not very well defined mathematical quantity. It depends on the environment, it depends on the conditions, it depends on the strains that you have, and so on and so forth. So we have few data, and it's harder to interpret this data compared to the structural problem. So this is why predicting function from sequence data or structured data is, for the moment, not solved completely. So we have a lot of work being done, and there is a lot of progress, but it's a hard problem. So what we try to do in our group is to develop methods to learn something about function from sequence data. We don't use structure, so we just skip structure and we go directly from sequence to function. And the reason why we skip structure is for computational simplicity, because structure is, I mean, we have powerful methods to predict structure, but they are computationally demanding. And so the models that we use are relatively simple, and including structure makes them more complicated and makes it more computationally difficult. I mean, it requires more computational resources to obtain results. So for the moment, we try to keep it as simple as possible, and we only use sequence data. But of course, we are working to include also structural information into our models. So this is essentially what we try to do. We want to learn something about the relationship between sequence and function. And we would like to use this to design artificial protein sequences that have desired functions, understand how nature does it. So nature is able to produce functional proteins by evolution. So we would like to understand how this happens. And in principle, we would also like to understand how we can do something like evolution in the lab. So people are doing a lot of experiments on this, and we would like to understand how to make models of these experiments. So we try to do this using data. So we use a data-driven approach, and we try to use models that are inspired to statistical physics. This is the main goal of what we do. So now, before I talk about the models, I can discuss what are the kind of data that we can use in our work. So there are many data on proteins. So I will try to show 4 main classes of data that we can use. So the first kind of data that we can use is natural protein sequences. So nature produced a huge diversity of sequences by evolution. So for example, if you imagine these kind of beta-lactamase proteins that bacteria use to develop resistance to antibiotics, these are very old proteins. So they have evolved for a long time. So there was maybe a long time ago some ancestral protein that then has evolved. And today we see many strains that are derived from this ancestral protein. Now, because evolution has been running for a long time, if you take 2 of these proteins that are, I mean, different sequences, but they do the same thing, so they fold into the same structure and they do the same function. But if you look to the sequence is very different. So typically, if you take 2 proteins of this kind, you have about 80% of the sequence that has mutated. So there are 80% of the amino acids that are different. And this is because evolution has been running for such a long time. So this is a very diverse set of sequences that are very different, but they all do the same function. So we can take advantage of this data to learn something about the constraints that are necessary to realize the function that we want to study. And in particular, we will be looking at 2 main features from this kind of data. One is what is called conservation. Yes?
Professor Zamponi, I'm wondering about the proteins. Should we think
Yes.
about them as you're saying they are from the same structure? Does that mean they come from the same organism, like the same environment?
No, 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 no.
Like,
No, yeah, this is important. No, they come from different organisms. So you have a variety of bacteria that can use these beta-lactamases to resist antibiotics. So you have different organisms. You have different conditions. So, for example, some of these proteins are typically derived from patients in hospitals. These are typically proteins that you find in bacteria that are very resistant to antibiotics. So people go to the hospital and they are treated, and you get this data from the proteins that they have in the bacteria that they have in their body. And some of these proteins, I will show examples, maybe were sequenced in some hospital in Sweden and some other in some hospital in the US or in Italy or in some other country. So this comes from very different, say, environments, and in principle from different bacteria. The thing is that they all do the same thing, so they all fold in a very similar shape, and they all do the same thing, which is cut the antibiotics. Okay, so this is why we think that they are a good this kind of data are a good way to learn about what are the minimal set of constraints that you want to have in a sequence to make a beta-lactamase protein, for example. Okay, so two things that are very important from this kind of data are what we call conservation, what people call conservation, and what people call coevolution. So conservation means that so each line here is a protein taken from a different species, for example, and you can align these proteins. There are there are tools that allow you to align these proteins and compare them. So you can look to a given site in a given position of the protein. And for example, you see here you always get a certain amino acid, for example, a D on this site. So this means that in order for the protein to function, you really need to have a D here. So this site is conserved. Even if the protein mutates, here you cannot do anything else than just put a D. So if you mutate this D, the protein doesn't function anymore. The other kind of features that we can get from this data are what is called coevolution. Coevolution means that, for example, if you have a V here, then you need to have a T here, you see, in all cases. And this means that, for example, this can be due to a contact. So in the three-dimensional structure, maybe this site is touching this site when the protein folds. And so if you make a mutation here, you have to compensate. So for example, if you have an amino acid that is bigger and another one that is smaller, if you mutate this one to a smaller one, you have to make the other one bigger. So you have this kind of interactions or correlations between sites. So, okay, so from this data, we can try to learn these things and try to learn a minimal set of constraints that we need to do to include to make functional proteins. And because these data are very diverse, so you have proteins that are very different, that have a lot of mutations, we represent it in this way. We say, okay, if we look to the space of all possible sequences, these are very far away, these sequences, they are very different. So it's like we have a space and we have points in this space that are very far away, and they cover more or less all the possible space of sequences. And we will give a score to these sequences. We'll say, okay, we will give an energy to these sequences. We call it energy, but it's not physical energy, it's just some statistical score. And we will say, okay, if the energy is low, this is a good protein, is a good functioning protein. If the energy is big, this is going to be a bad protein. So we will try to construct a model that gives low energy to all the natural sequences because these are proteins that nature selected to work well. Okay, so this is the first kind of data that we are going to use. Now, the second kind of data that you can use is what is called deep mutational scans. So deep mutational scans are experiments where you take one protein that you take from, I don't know, one bacteria, you take one protein, and then you take all the possible sites in the protein and you make all possible mutations on, on each site. So for example, if you have this protein, so in a typical protein, you will have about 100 or 200 sites. So say you have 100 sites. On each site, you can make 19 mutations because you have 20 amino acids. So there is one amino acid here, but you can substitute all the other ones. So you will have to test, if you want to test all the single mutations, you will have to test 1,900 possible mutants, okay? So people are able to do this in the lab, and so they will measure for all the possible single mutants whether it functions or not. So they will take the protein, they will put it in bacteria, and they will measure if the bacteria is able to survive to antibiotics or not, and in this way they can assign a fitness to each mutant. When they do this, you have let's say you have one single sequence in the space of sequences, but you are looking to all the possible neighbors, all the possible sequences around it. So you are able to know around a given sequence what is the shape of the fitness space, okay? So these are data that are useful to check, for example, if your model is able to predict this kind of things. The next kind of data that we can use, which are the main focus of our work, is what is called in vitro evolution experiments. So these are experiments where you start with a certain protein that you, again, it's like some natural protein. You put the protein into bacteria, but you do, you create a library of mutants of this protein. So for example, you take the protein, you do PCR, so you make copies of the proteins, but you introduce errors. So maybe each of your copies will have maybe 1 or 2 mutations, 1 to 3 mutations, a few mutations. You put this library into the bacteria, you grow the bacteria in presence of the antibiotic, and then some bacteria have good mutations, so they will survive, and some other bacteria have bad mutations, and they will die. So you select the bacteria that survive, you take out the DNA of the protein, and you do it again. So you make other copies with mutations, and you do it again and again. So you do a few cycles of this. In this way, you can accumulate mutations, and you can go a bit, you can go far away from the original sequence and introduce mutations and try to preserve the function of the protein. So in these experiments, so this experiment has been performed by a group in the US. This experiment has been performed in Italy, and this experiment has been performed in Canada. So there are 3 different groups from 3 different countries did this kind of experiments independently, and typically they can do between 20, 30, a bit more of these cycles, because it's very expensive and it's very time consuming. And they end up with mutants that have maybe 10, 20% mutations with respect to the original protein. So you start with the protein and you are able to mutate about 20% of it. And then you can sequence the mutants, and you have a lot of sequences. The good thing about these experiments is that they are done in the lab, in vitro, in a lab. So you have very good control of the environment, and you can sequence a lot. So in the end, you have 10 to the 5, 10 to the 6 mutants that are sequenced, and you have the data. So this is like, in this representation, this is like you start from a natural sequence, and then you grow a cloud of mutants around it. But you don't go very far, because, as I told you before, if you look to natural evolution has been running for many more generations. And so as a result, your typical natural proteins are much more distant in the space of sequences. So you have 80% of the protein that is mutated, while here it's only 20% of the protein that is mutated. So you can imagine that natural evolution was able to explore much more this space and reach faraway regions that you don't see in these in vitro experiments. Okay. So these are the kind of data that we have. There is a last group of data that we can also have. These are experiments where people take, for example, 2 natural sequences, and then they construct all the possible intermediate sequences. So, for example, consider this experiment. This is a very beautiful work in which they took two proteins, this protein here and this protein here. So this protein is fluorescent in the blue, and this protein is fluorescent in the red. So what they did, and okay, these 2 proteins differ by 13 mutations. So there are 13 sites that are mutated. So since you have 13 sites, and each site has 2 possible amino acids, one in this case and one in this case, what they did in the experiment was to construct all the intermediate states. So you have 2 to the 13 proteins that are in between, which is 8,200 proteins, more or less. And for each of them, they could measure the blue and the red fluorescence. So what they find is that most of the mutants, most of the intermediates are not functional. They are not fluorescent, not in the blue, not in the red. Some are fluorescent in the blue, and some are fluorescent in the red. And so you have this kind of bottleneck shape, where you have here a lot of blue proteins that are obtained by taking this protein and make 1 or 2 or 3 or 4 mutations. At some point, when you make about 7 mutations, you can switch the fluorescence from blue to red, and then you have the other group that is fluorescent in the red. So this is like you start from here the blue protein, here you have the red protein, and you construct a path of mutations that bring you from one to the other. And this is a similar work where they studied antibodies. These are antibodies that bind to the strain of the flu virus. So these antibodies can bind to different strains, and they considered all the possible mutations between the original, say, antibody and the one that was mutated. And they measured the ability of all the intermediates to bind. So they have a similar kind of data. So there are more and more experiments that are appearing about this, with this kind of data. And so it is interesting to have models that can tell you how this space of intermediate proteins is organized and how you can mutate from one to the other, okay? So these are the kind of data that we try to use in our work. And basically the take-home message on this of this part of my talk is that there are many data that are appearing because people are able to sequence much more. It's becoming cheaper, it's becoming easier, and so there are many data, and we need models to make sense of all this data. Okay, I don't know if you have any questions up to this point.
Yes, Professor. I have a question about the previous slide. The diagram B that is showing the mutational steps, and I think the log.
Sorry, I cannot hear you very well. Can you? Yeah, maybe.
Okay.
Yes, this is better.
Okay. I was asking about this very slide about this diagram B, the graph, about mutational step mainly. I want to. Should I think about the mutational steps in form of like time as the evolution, or as time happens, this particular sequence evolves to another sequence that has transformed into, or that it has evolved to. So the steps that happen in the process of mutation, should we think about them as time, or sort of like changes in the sequence itself?
Yes, I understand. So this depends a lot. So this is a difficult problem, I think. So in this experiment, there is no time. They took two proteins, they do all the possible mutations in between the two, and they check if they work or not. So this doesn't tell you anything about how. So if you start evolution here and you wait and you let the time go, we don't know what would happen in this case. I mean, we don't have an experiment here. This is just a static snapshot of what is the fluorescence of each protein. But then if you start evolution here and you run it and you wait, you will have a random process. So maybe some mutation will appear and there will be some path in this space, but we don't know which path will happen. So this is, maybe I'm going to discuss a bit later about this. In this experiment, there is time. So because this is an experiment on antibodies, so they have a patient and they know that they took this antibody from the patient. This is the somatic one. And they know that this patient at the beginning had this germline antibody. And so here we know that there is, over time, the patient was able to develop an antibody that has a good binding to this virus. And so you can try to reconstruct a time process that brings you from here to here. Okay, so here there is a time, and in the paper, if you look to the paper, they try to reconstruct, to infer the time evolution of this antibody. And this is another example. This is a paper where they took some protein, and they took the protein that we observed today, and they tried to reconstruct the ancestor of this protein. And then they went to the lab and they made an experiment to test the functionality of the ancestor and try to. And so here again, there is a notion of time. So this is like natural evolution, and you can try. But the problem is that you always have to infer the time evolution because you don't observe it in these experiments. You just try to reconstruct the time evolution. In this kind of process, instead, you can observe the time evolution. So you start from a bacteria, you make mutations. So you can say, okay, there is the first generation is this one. Then there is the second generation, and you can measure it. Then you go to the third generation and you can measure, and you can observe how the evolution happens with time. Okay? So I will try to explain how we can construct a model of this evolutionary process over time and compare with the experiments. Okay?
All right.
Okay, thank you for the question. So how do we construct the model? So we want to construct a model which is a stochastic model where we have a protein, we make a mutation, and then we accept the mutation or not. So now I will try to explain how we try to construct a function that we can use to decide if we accept the mutation or not. So to do this, we make use of what is called generative modeling. I think you know how it works. But so let's see very schematically. Generative modeling means that you, for example, you start from many images of human faces, and then you construct a model that is a probability distribution over the space of images. And so then you extract new images from this probability distribution. And if your model is good, the new images look like human faces. So like this one. So this is a person that doesn't exist, and the model generated it. Very similarly, you can do the same for text. So you take a lot of text, you create a probability distribution over text, and then you can generate random text that, if you are good in your modeling, looks like good text. And this is what, ChatGPT and similar machines do. So we try to do the same thing for proteins. So we start from data, and the data are many proteins that are the natural ones, for example. So we take this kind of data, we take natural protein sequences that are aligned. They are all equal in structure and in function, but they have different sequences. So we show to the machine all these sequences, and we build a probability distribution, and then we can use the probability distribution to generate new sequences. So how do we build the model? So in order to build the model, we try to use very simple machines that are called Boltzmann machines. So Boltzmann machines are probability distributions. So you have a sequence of letters that are the letters of your protein, of the protein sequence. And the Boltzmann machine is a very simple probability distribution over the letters, which is the exponential of a sum of terms. You have a sum of terms that are one for each letter. So you have a term for the first letter, a term for the second letter, and so on and so forth. And this can be used to model conservation. So for example, if you really want to have a D here, you will put a term here on the third side that gives a very large probability to put a D and zero probability to all the other letters. So in this way, you can represent this kind of information. The other term that we put is a pairwise term. So we put a sum of terms that depend on two sides. So for example, if we want if we know that every time that there is a V here, there is a T here, we can put a term here that if there is a V in the first side, will give a very high probability to a T on the other side. Okay? So this is a minimal model that has a large number of parameters, but not so large. So if you have 100 sides, this will be about 100 terms, and this will be about 100 square terms. So you end up with, about 10 to the 4 parameters, which is not huge. And you can train these models relatively easily on a machine. So our models are simple, but they are energy efficient in the sense that you can train on your computer. You don't need a supercomputer. So you don't need to spend billions of dollars into huge data centers to train these models. You can just do it at home. So this is why we like these models, and we try to, I mean, we think it's important that these models can be used by everyone. So we also, as I said, we have codes that we can share to train these models and use them. So this is an example of a test of the generative properties of this model. So this is a work that appeared in Science 5 years ago. I was not involved in this work, but my colleague Martin White was involved in it. So what did they do? So this is a protein that is called chorismate mutase. It's not the beta-lactamase, it's another protein. So chorismate mutase is an enzyme, and this enzyme is used by bacteria to produce certain amino acids. So that they need to function. So if you put your bacteria in a medium without these amino acids, the bacteria have to use this enzyme to produce the amino acid and survive. So if this enzyme is not functioning, the bacteria will die. So then what do? They take natural sequences. So this chorismate mutase exists in many different species. So you take all the natural sequences from different species and you build a model. So then what you can do is, in parallel, you can take the natural sequences from many species and you do an experiment where you put them into E. coli bacteria and you let them grow. If the E. coli is able to use the enzyme, it will survive. So what you see here on the horizontal axis is the growth rate, essentially, of the bacteria. So these bacteria were able to grow because they can use the enzyme, because the enzyme works. These bacteria here, they don't grow, they die. Why? Because they are not able to use the enzyme taken from another species to grow. This is because when you take a protein from a species and you put it in another species, sometimes it doesn't work. I mean, the E. coli is not able to express this protein or to use it and so on and so forth. So if you take natural data, about 30% of the proteins work and the remaining do not work. Now, these are natural sequences. Now here you have artificial sequences that were generated by the model. Okay, so they trained the model, they use the model to generate new sequences, and they see if they work. And what you see is that, again, about 30% of the sequences work, and the rest doesn't work. So this means that your model is good, is working well, in the sense that it is able to generate sequences, artificial sequences, that have the same probability of functioning compared to natural sequences. You should keep in mind that the model is able, so we can estimate the number of different sequences that the model can generate, and this number is huge. So the model is able to generate about 10 to the 65 sequences. And so this is a huge number. In the lab, you can only test a few thousand sequences. But in principle, the model can generate many more. So this is a proof of concept of the fact that the model works and that these two ingredients, conservation and coevolution, are the minimal ingredients that you need to make it work. If you remove coevolution, for example, if you only make a model with conservation, it doesn't work. You don't get any functional sequence. You have 0% fraction of success. So this is the summary of this part. So we can use generative models to generate artificial proteins. You can do experiments and it works. These are a few references that you can check if you are interested. You can use Boltzmann machines as we do, but you can also use different type of models, like, for example, generative adversarial networks, variational encoders, and so on. So this is there is a freedom in the choice of the architecture. What we are doing now, so first of all, as I said, we have packages that can be used on the GitHub. So we are trying to optimize the codes and make them as efficient as possible. We are trying to develop procedures to reduce the number of parameters of the model as much as possible. So you can check this work if you want. And we also have a test on not on proteins, but on RNA, where we can generate artificial RNAs that are functional, and we can compare the different methods, the Boltzmann machine, the deep networks, different architectures, and we find that in general the Boltzmann machine is good enough. It's not worse than other models. So it is simpler, but it is not bad. So if you want, the take-home message for this part is that simple generative models can design artificial proteins, and so we can generate a diversity of many different sequences that will work. Now, the next step, as I was saying before, and you were asking, is can we use this to understand how things will mutate over time? Okay. So is there any question on this part?
I think there is no question.
Yes.
No, you can proceed.
Okay. So I will move to the next part. So the next part is to try to study dynamics, so time evolution. So what do we do when we have, so now we have a probability distribution. So using this model, we have a probability distribution, and high probability means the sequence is functional. Low probability means the sequence is not functional, it's not a good sequence. So what can we do now to make a model of evolution is to use what is called Monte Carlo sampling. So Monte Carlo sampling is the following thing. You have a probability distribution. You start from a sequence. Let's say you start from a natural sequence that you have in your data set. And then you propose a mutation. So you say, okay, maybe I take this amino acid and I change it. And what you do, you accept the mutation with probability P given by the model, or otherwise you reject it. So if the mutation brings you to higher probability, then it is probable that you accept it. If the mutation brings you to low probability, you will probably reject it. So you do this many times, and you can try to use this as a model for evolution. Now, so let me try to explain now the whole pipeline of what we do. So let's say you want to make an experiment in the lab where you have a protein and you want to evolve it and see what happens. For example, because you want to evolve it to develop a new function, or maybe because you want just to see how it can mutate. So this is your initial sequence, the protein that you are interested in. So what we do, the first thing we do is to do what is called homology search. So we look for proteins, for natural proteins that are similar to this one. We construct a data set of natural proteins that are similar to this one. From this, we can learn the generative model that I was discussing before. And then we can do the Monte Carlo procedure. So we take the protein, we propose a mutation, and we accept the mutation with the probability that is given by this model. And we do it many times, and we see how the mutations accumulate or not. Now you can say, okay, maybe this will work, maybe this will not work. Why? What is the problem? Why should it not work? The problem is that we learn the probability to accept the mutations using natural sequences. Now, natural sequences have been produced by evolution over billions of years in very different conditions. Maybe the environment was warmer or was more humid, or the species were different, and so on and so forth. While the evolution in the lab is done in very controlled conditions and that are maybe different from the natural ones. So we are trying to learn the probability of accepting a mutation from natural data and to use it to describe something in principle different. On top of that, we know that in natural evolution there are phylogenetic effects. So things evolve on a tree, there is a phylogenetic tree, and so the probability that a mutation is accepted or not depends on the size of the population, on the way that things have evolved, and so on. While in our model, we only consider independent chains. So we take one protein, we make mutations, and then we repeat it independently, okay? So there are many things that are different. So the question is, does it work or not? So in order to check if it works, we compared our model with experiments. So there are two experiments. As I was saying before, there is this one. This is done in the US. They started from this protein. The protein is called PSE1, is a beta-lactamase. It is one of these proteins that give antibiotic resistance to bacteria. And they did mutations on it, and they measured and they sequenced the mutants. And the same thing was done in this paper from an Italian group, using another protein, a very different protein that belongs to the same family. So it does the same thing, but it's a different protein. So I will maybe skip. Okay, I will go to this one directly. So we can use the model, and so we can compare the library of the mutants that have been generated in the experiment with a similar library of mutants that have been generated in the computer by our model, okay? So how do we compare them? So in order to compare them, we do the following thing. So this is the protein. The protein is a protein of 202 sites. So here you have all the sites in the protein that are labeled along the sequence. On each site, we can look in the experiment how many mutations you get on that site. In order to quantify the number of mutations that you have, we use a quantity that is called, it's called entropy in information theory and in physics. So for those of you who have a more like information theory background. But in any case, what you get here is essentially how many mutations you can have. So when this number is small, it means that this site cannot mutate. When this number is big, this site can mutate. So what you see, the purple line is the experiment, and the yellow line is the model. So what you see is that we can predict from the model which sites are more likely to mutate and which sites are not going to mutate, okay? Furthermore, we have two experiments. So with the same model, we can model the two experiments. And you see that there are some sites, for example, let's see, there are some sites that are more mutable in one experiment, like for example, this one. This site is very mutable in this experiment, and it is not very mutable in the other experiment. Why? Because the starting point in the two experiments is different. So the two initial proteins are different, and then this protein is going to tolerate a lot of mutations here, while this protein is not tolerating mutations here. And the model is able to tell you that. So the model is able to reproduce this thing. So So the message of this part is that you can use these models to make predictions on which sites will mutate on in your experiment, and how much they can mutate depending on the initial protein that you consider. Okay. I will show you one last thing, and then I will conclude, which is, I will skip this part, okay, which is this figure. So this figure is taken from this paper that we published recently, and it gives you the following thing. So on the horizontal axis you have time. Okay, this is Monte Carlo steps. So each Monte Carlo step we propose a mutation, and the mutation is either accepted or not. So the more mutations we propose, and the more time is passing. So this is more or less time. And on the vertical axis you have how many mutations you can accumulate on a given site. So each line is one site in the protein. There are about 100 sites, is another protein. No, sorry, it's the same protein. So there are 200 sites. And what you see is that there are 3 different kinds of sites. There are the green sites are sites that are conserved. So these sites cannot mutate, and they will never mutate, basically. So the number of mutations is always going to be very, very small. These sites will never mutate. Then there are the red sites, that these are variable sites. So these are sites in the protein that are very easy to mutate somehow. So when you run your simulation, very quickly, these sites will mutate, and you will see a lot of mutations. And then it will stay like this when time goes. But the most interesting feature is that you have some sites that are called epistatic. So these are sites that can only mutate if some other site mutate, because they are correlated to other sites. So at the beginning, they will not mutate because the rest of the protein is fixed. And if you try to make a mutation, the mutation is going to be rejected. It will kill the bacteria. You have to wait a lot of time. If you wait a lot of time, other sites maybe will mutate, and then it will allow your site to mutate. So these are the blue sites, and you see that you have to wait a lot of time before they mutate. And they mutate in a very slow way. You keep accumulating mutations until you reach the long time behavior. So this is a figure that was constructed using the computer simulation with our model. But experiments, for the moment, can produce data about this time scale. So we can compare the model. I mean, this comparison of the model with experiment, this one that I showed before, was done at using the longest experiments we have, that are more or less here. So in the experiments that people can do today, you cannot see these long time scales of mutations. But maybe in the future, we will have longer experiments that maybe are able to reach this regime. Natural evolution has been running for much longer, and it is probably somehow exploring this regime. And in principle, our model can be used to interpolate the 2 regimes. And so this is my final message, that the evolution with time of the sites is very heterogeneous. There are sites that can move very fast, that can mutate very fast, and there are sites that can only mutate after much longer time when other sites have mutated. And in principle, the model is able to give you some prediction of this information. So I will skip this. I just want to mention that if you are interested in these techniques, we have a book that I wrote with two colleagues, and if you want, I can send you a copy of this book. So if you are interested, let me know. And with this, I thank you very much for your attention, and I'm happy to answer any questions you might have.
Thank you very much. Thank you very much, Professor Zamponi, for the interesting talk. And unfortunately, the colleague who is currently tackling physics-based, I think with some sort of statistical mechanics problems, as a doctor, I think is on blacked out. For the book, I think he will find it interesting, but also even other members who are not here. So we would be honored, Prof, if you provide us with a copy. I don't know whether I'm audible enough.
Yeah, if you send me an email, then I can find a way to send you a copy of the book.
Yes, I know some members were running to classes. So I don't know whether there are questions in the audience. For me, Prof, in the meantime, when I think about generative models, my bias like takes me to diffusion models, and as you guided the talk, it is interesting to see what diffusion models would produce at a sequence level when applied to mutations.
Yes.
So,
yes, I agree.
My intuition is to really, if we can pick up the work we've done and look at what diffusion models could produce or give us. That's what I've been thinking as we are presenting.
Mm-hmm. No, I agree. I mean, what we have, some people in our group are trying to do something like this. You take your sequence, you represent your sequence in an abstract space by time encoding, and then you try to do diffusion in this space and see if you can model the evolutionary process on the sequence by a diffusion in this representation. And it's a very interesting idea. So I think it's something we should think about and try to see if we can make a good predictions out of it. So this would be really cool.
Interesting. Also, last year, a lot of physics-based machine learning papers came out, and maybe in 1 minute, I don't know, you could maybe summarize for us or tell us about the physics-based aspects we can pick from this work that generative modeling.
Yes, sure. So you mean in another session?
No, I was referring to this very session, like the takeaways in form of physics-based machine learning in regards to this work.
Okay, so in this work, basically, let's see. The advantage of, so the models that we use, this kind of model is a physics-based model. It's what is called the Boltzmann distribution in physics. And I think what is interesting from the physics point of view is that when you use this kind of models, you can very easily compute what is the entropy of your model, which in physics we use a lot, and it's easy to compute for this kind of models, while it is much more difficult to compute for other generative models. And the entropy tells you how much diversity you can generate from a given model. And so the higher the entropy, the better your model is at generating a diverse set of things. And what I think from the physics point of view, it's interesting that we can estimate the entropy, and we see that the proteins, these models can generate a huge number of proteins that are functional, much more than natural evolution can explore, which means that nature has found a few proteins that work well, but there is a huge space of proteins that work equally well. And in principle, using these models and the idea of entropy, we can quantify how many proteins you have that, in principle, that do something. And this tells you also something about what is the probability of finding one protein that works. So we have been working a little bit in okay, let me see, in this paper here, in these two papers, on, for example, estimating the entropy of the space of some RNAs that are able to self-replicate. And the idea is to try to understand what is the probability to find, by chance, an RNA that self-replicates. And so what is the probability that you can build life from RNA, for example. And so I think, the only thing I think is interesting is that I think entropy is a very important concept from physics. That is, entropy comes both from physics and information theory, but it is used a lot in physics. And it gives you some way of quantifying diversity. And it's important because when people talk about generative models, like of faces or text, it's very hard to understand how much diversity your model can generate. And in principle, in these physics-inspired models, we can do it. So I don't know, there is something there. Now, there are a lot of other works on the interface between physics and machine learning is very big. So I cannot, there are many things that one could say. If you want, if you're interested in this, I can maybe give you a few names of colleagues that you can invite that work on statistical physics and machine learning, and they can give you an overview of the research that is being done, at the interface between statistical physics and machine learning. But I think for our work in particular, bringing this notion of entropy into the field is something that can be useful for understanding some properties of the evolution, let's say.
Thank you, Professor.
Thank you.
Definitely, we intend to have more talks within physics-based machine learning. So we will be honored, Prof, if you connect us to the researchers within that area. I think I'll structure the requests in an email that I'm going to send to you, plus the request for the book. So we've reached the top of the hour. I want to thank everyone who has attended.
Thank you very much.
And thank you, yes, Prof. Gaboni, for giving us this talk.
Thank you.
So we'll be uploading it tonight on our channel, and yeah.
Okay,
to the
great.
rest of the members, thank you. See you in the next session next week.
Thank you very much. Bye-bye.
Bye-bye, everyone, and bye-bye, Prof.